Hello, I'm Professor Paul Bingham, and this is Biochemistry 1. Our goal in this segment is to take the first steps toward mastery of the second half of the citric acid cycle. This is the second of a two-segment set on this cycle. So if you haven't recently watched Citric Acid Cycle 1, you should have that fresh in your mind before beginning. The overall thrust of this, you'll recall, is of this set of segments, is to understand how the macromolecular machines that serve as catalysts made by gene products for the purpose of allowing the successful life and replication of organisms, vehicles, oak trees, human beings, bacterial cells, any organism. An important subset of those has the goal of fluxing fuel through our bodies, a lot of it, very high rate. So all the food we eat each day, energy is extracted from that to allow us to move around the landscape, and matter is extracted from that to build spare parts to replace the worn out bits, so to speak, of our bodies as a result of our everyday lives. That process, that flux of matter and energy through our bodies at very high rates, very large amounts, collectively is referred to as metabolism. And we've been looking at that process in the last several segments. We'll continue to look at it in the next several segments. Remember the overall thrust of this. We take in reduced hydrocarbons. Food is, from a biochemist's point of view, reduced hydrocarbons. And we oxidize those hydrocarbons, ultimately to CO2 and water, extracting large amounts of energy in the process. And the TCA cycle is central to this entire metabolic picture. The acetyl-CoA derived from glycolysis, but also from any other source, from fatty acid degradation, from amino acid degradation, all fluxes through this TCA cycle or citric acid cycle or Krebs cycle as it's variously called. So this relatively simple 8-step cycle, in other words, stands right at the center of all of the energy metabolism that makes all organisms, at least all aerobic organisms around the planet, again, oak trees and human beings included, go, make them run, lets them run. So let's dive in where we left off last time. That is at the fifth of 8 steps in the citric acid cycle, the succinyl-CoA synthase cycle. So remember that we left off last time with the alpha-ketoglutarate dehydrogenase step, which we again looked at the end of the last segment. We're now going to skip to or move ahead to the next step in the cycle, the succinyl-CoA synthase step. Remember now, to avoid confusion, this enzyme is named backwards, as if its purpose were to catalyze the anti-TCA cycle rather than the forward TCA cycle. So don't be misled by its name. Let's look at what it actually does. Notice that it's releasing CoA from succinyl-CoA. So it's hydrolyzing that high-energy thioester linkage between succinic acid, succinate, and acetyl-CoA, and it's turning that into phosphate anhydride on GTP. So that's a cool trick, and now let's look at how you can actually arrange to do such a thing. So let's zero in on the reaction mechanism and look at it carefully for a moment. So notice that the first thing that happens is the enzyme positions succinyl-CoA and a free organic orthophosphate in a proper position for nucleophilic attack by one of the phosphate oxygens on the carbonyl carbon, or the carboxylate carbon of succinyl-CoA. This allows phosphorylasis of the succinyl-CoA product and produces the succinyl phosphate product, still bound to the enzyme, shown at right here. So we don't normally call succinyl phosphate part of the Krebs cycle because it's an enzyme-bound transition state, if you will, or a reaction intermediate. The next step then is for an enzyme histidine to attack the histidine ring nitrogen to engage in a nucleophilic attack on the phosphate of that succinyl phosphate group, capturing the phosphate in a high-energy phosphohistidine intermediate and releasing free succinate, the first product of the reaction. Then in the third set of steps, the oxygen of a GDP molecule is positioned to engage in nucleophilic attack of the phosphorus in the enzyme-bound, covalently bound phosphate histidine intermediate, displacing it from histidine onto the GDP attacker, creating CTP, and storing that high-energy bond in a phosphate anhydride linkage cell. a sort of nifty set of steps here, where there's a covalent intermediate, enzyme phosphate intermediate, captures this free energy from the thioester bond, converts it into GTP, high energy anhydride bond. Nifty trick. Okay. Notice one other thing before we leave this. This molecule, succinate, is the first fully symmetric molecule since citrate in the TCA cycle. I'm sorry, rather since isocitrate in the TCA cycle. And so any previously existing chiral centers have now been obliterated. So as we understand how carbon flows through the TCA cycle over multiple cycles, this will turn out to be important. You've erased any chirality here. You've effectively randomized the remaining 4 carbons on this intermediate. We'll call that back in a few minutes because it allows us to make sense out of what happens to carbon overall. All right, let's go to reaction 6. Remember, there are only 8, only 2 more to go after this one. This is the succinate dehydrogenase reaction. So here's the succinyl synthase reaction. We're now moving up to succinate dehydrogenase. Succinate dehydrogenase. Notice what it's doing. It's now producing not a reduced NAD, as several of the earlier enzymes did, but it's now producing a reduced FAD. Remember what FAD is? It has all the nucleotide paraphernalia of a lot of these coenzymes, but its business end is this flavin ring system, which can accept either 1 or, in this case, 2 electrons, producing FAD-H2, as it's called, the fully reduced FAD coenzyme here. There's something a little unusual about the FAD in succinate dehydrogenase. This coenzyme is now actually covalently bonded to a histidine residue, as diagrammed here, in the active site of the enzyme. That's not normally the case. In the case of the E3 subunits of PDH and alpha KDH, for example, the FAD is non-covalent, tightly but non-covalently bound. In this case, it's literally covalently bound. An interesting wrinkle on this particular enzyme. All right. So here is that reaction. So enzyme-bound FAD is reduced by abstracting reducing potential, that is oxidizing succinate to fumarate. So notice here that the succinate dehydrogenase enzyme interacts with the substrate stereospecifically, so the fumarate ends up specifically in the trans configuration across the introduced double bond as indicated here. What that means is, now having erased chirality with succinate, you're now creating the potential for new chiral centers moving forward from here. Okay? Reaction 7 in the cycle. So we just looked at succinate dehydrogenase. We're now looking at the fumarate reaction. And fumarase, or fumarate hydratase as it's sometimes called, introduces a water molecule essentially rearranging across this double bond, rearranging to create a malate in preparation for the next reaction. So let's look at that fumarase, or fumarate hydratase, that catalyzes this reaction. Notice what's going on here. A water is being inserted across this double bond to turn it into a single bonded molecule. Notice also that there is now a chiral center here, circled in green. That chiral center will now be stereospecific again. Again, let me remind you of why that is. Because of the way enzymes interact on an atom-by-atom basis with their substrates, 2 stereoisomers of the same chiral molecule are apples and oranges to an enzyme. They're completely different molecules. As difficult as they are for us as organic chemists to distinguish, for an enzyme, they're quite easy to distinguish. And when there is a chiral center, enzymes always care which stereoisomer is involved, and a specific stereoisomer of malate is introduced here. So let's complete the cycle, getting all the way back to the beginning. So we just looked at the fumarate, or fumarase, or fumarate hydratase. Let's now look at malate dehydrogenase, the last step. That's going to generate another reduced NAD. So remember what we're doing here. We took 2 carbons in at the beginning in the form of acetyl-CoA. We've released 2 carbons as CO2, so we're in the business of gradually oxidizing those 2 carbons. We don't oxidize them, all of those 2 carbons in one cycle. It takes several cycles to oxidize them completely, and in fact this is another of those oxidation steps where we reduce NAD, taking reducing potential away for use.